Good afternoon, everybody. It's my great pleasure to introduce Dr. Patrick Keeling to present his Storer lecture here at UC Davis. First, I'd like to say a few words about the Storers whose generosity has allowed us to bring world-leading scientists to campus and has done so for the last 60 years. So it's already, the Storer Lectureship has a long history at UC Davis. And this year, it's particularly poignant because it marks the 100th anniversary of Tracy Storer actually arriving on campus in 1923 as the first faculty in zoology, which later became the College of Biological Sciences. So he's really a founding and historic member of the UC Davis community. His pioneering work on biodiversity in Yosemite broke new ground in the field of ecology. And his textbook, General Zoology, became a core reference text in the discipline for decades. And his wife, who also is a named member of this lecture series, Dr. Ruth Storer, was a trailblazer in her own right. She was the only female graduate in 1913 in medicine from the University of California Medical School, which is now UCSF. And she was the first female physician in Yolo County, and she had a pediatrics practice that she maintained until she retired at the age of 80 years old here in Davis. And they both really had a true passion for learning and for disseminating knowledge, and that lives through with this lectureship, and I imagine that they'd be excited to see Patrick's lecture today, and we're really honored to be joined by Ian Storer, who is a member of the Storer family and continues in that tradition with that passion for knowledge. So that brings us to Patrick. So Professor Keeling is a native of Ontario, Canada, and he did his undergraduate degree at the University of Western Ontario, followed by a PhD at Dalhousie University under Professor for Doolittle. Sorry, I lost my place. And he studied the prokaryotic and eukaryotic transition. And he has his own lab now at University of British Columbia, where he's made numerous advances in assembling the eukaryotic tree of life. And really, due to his work, every couple of years, the field has to either add or make a significant change to the eukaryotic tree. And he's really, shown us that, how far we've come in our understanding of biodiversity, but also still how far we have to go to really understand the biodiversity of eukaryotes, especially of the single-celled eukaryotes, the protists. And for his work, he's received many awards, including the Gilbert Morgan Smith Medal from the National Academy of Sciences. He's the most recent recipient of that in 2021. He's highly regarded in his field. And when I mentioned his visit to our colleague, Professor Jonathan Eisen, who regrettably couldn't be here in person today, he remarked, quote, "I honestly consider him one of the greatest scientists I've ever encountered, and I'm truly and deeply sad I will have to miss this." So without any further ado, it is my pleasure to present Dr. Patrick Keeling for the Storer lecture.
All right, can you hear me here? Okay, I'll just do this then. It'll be kind of like a... All right. So thank you very much again. I appreciate the invitation and the kind words. That's great. So today I'm going to give a lecture sort of at a more... I was asked to do something philosophical. I don't know if I can do that. But I will try to talk about more general things about eukaryotic evolution that have to do with how selection works, how everything changed is the point I'm trying to make. When eukaryotes evolved, it changed so much at so many different levels. And I'm going to use one topic, horizontal gene transfer, mostly as an example to make the point that I'm trying to make. And I gave this the title, How Eating Changed Our Genes, and that will make a little bit more sense later on, because that's one of the important things that eukaryotes do is eat other cells. But I want to actually start with a couple of discursions that are about how we show these things, the pictures we draw to try to illustrate these things, and why they're kind of shooting us in the foot. So this is a Google search of eukaryote versus prokaryote, which you heard was the title of my thesis. And you get these images that we might use for teaching or outreach about what's the difference between eukaryotic and prokaryotic cells. Okay? They're all very similar. I'm just going to take the top Google hit to show you what I mean. Prokaryotic cell, eukaryotic cell. These images are almost without exception terrible. They're full of mistakes. They're full of oversimplifications. They're just wrong. So there's all sorts, I won't go into them all, but there's numerous errors. Even in this simplified diagram, there's things that are false. The worst one that always gets me is that there's only one membrane around the bacteria, and most bacteria have two, and that has huge implications when we look at the biology of these organisms. The eukaryote, I don't even want to talk about it. It's just so awful. But there's a couple of things in particular that I want to draw your attention to. Well, I hate these pictures for two reasons, as the subtitle says. What are the two reasons? The first one is eukaryotes are not a blob. They're always drawn as a blob. It's like an animal cell that's been plucked from some tissue or something like that. This really bothers me as a eukaryotic microbiologist because eukaryotes are amazingly diverse and beautiful and structurally complex. So a couple of years ago, 13, I had this I don't know why this went dark, but I had this undergrad working in my lab, and we decided to draw a whole bunch of pictures of different kinds of eukaryotes for like textbook quality technical diagrams, like you'd want to see in a guidebook so you could identify them. And we just went through and just all these different kinds of eukaryotes, and we drew all these technical diagrams with all the different things labeled. The idea is these should be publicly available. They could be used for teaching or outreach. Instead of just showing a blob, you can actually show actual cells and what they sort of look like and all the important characteristics that they all have. And this kind of illustration has so much power to actually get across to people what these things are really like, what they're really made of, how they're built, how complicated they are, how you have this crazy non-symmetrical complexity in these cells. It's just astonishing, and this is what makes eukaryotes so cool, and we're missing it with these blobs. So these are actually going to come out shortly, and we picked an open access journal. We're going to distribute them as broadly as we can with a Creative Commons license so people can use them and adopt them and change them and whatnot. And we really would love to see more of this kind of thing because these images are so important to get across such an abstract, weird little world that we really don't understand. We put it all in the context of a tree with thumbnails, also make all this publicly available, so you can actually say something about how this blobs are not blobs. They're actually crazy, diverse, complicated cells with really crazy developmental programs that we don't really know anything about. So that's my first discursion. The second one is, these are filled with so many mistakes that it's undervaluing how big of an event the eukaryotic-prokaryotic prokaryotic-eukaryotic transition was. They're both oversimplified, and it doesn't really do this question justice. So I also have been working on actual pictures of, well, first of all, what's a prokaryote? It's a bacteria or an archaea. They're not the same thing. They're totally different, but you almost never see all three of them together. And then is a eukaryote really a blob? No, it's not a blob. It's a complex, non-symmetrical thing with feeding grooves and flagella and blah, 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 blah. Lots of stuff. So I've been working a lot on these kind of things lately. And I show this because this is what I want to talk about, is what makes eukaryotes so weird and different, and in my view, special. You can call me an essentialist. I'll wear that badge. That's fine. Because I think there's something about them that makes them different. And that's what I'm going to try to talk to you about today. This transition, when this happened. And these kind of cells evolved from these kind of cells. Okay, so to get at this, I want to first talk about ecology just slightly. This is, again, just a sort of an image that you sort of see for eukaryotic ecology, or for prokaryotic ecology a lot. It's basically a bunch of bacteria floating in the ocean, and it's basically they're forming a metabolic network. So the different bacteria are contributing different things to this network. They're interconnected through the metabolites that they're exchanging and whatnot. And this isn't a perfect representation of bacterial ecology, but it's actually not a bad representation of a lot of bacterial ecology. This kind of thing does happen a lot. And one of the reasons we do this is because we're very good at inferring this from genomes. You can look at a genome and you can see metabolic enzymes, and more often than not, you're right when you guess what they're doing, and then you can plug them into a network, and it kind of makes sense. The eukaryotes aren't in this, although I noticed a little while ago there is one. What's it doing? Photosynthesis. Oh, it just kills me. But anyway, plugging eukaryotes into this is actually hard, and I'm almost glad they didn't, because it would have been a nightmare. Because this kind of metabolic network isn't really what they do. Think to yourself, if you were to sequence the genome of this eukaryote and infer that it was highly intelligent and it could use tools to solve problems to find food, could you do that from the whole genome of this eukaryote? If you sequence that thing's genome, my assertion is you wouldn't know that it had feathers and could fly, let alone that it was smart and could use tools, unless you already had a bunch of genomes from other birds and hundreds of years of natural history to tell you what birds were and what they were like. Because the genome wouldn't tell you any of that. The genome would be a mess of unknown orths and things like that. Because the bird is a sum of a complex series of developmental pathways that give it structure, and the structure leads to function, which is behavior. And that's why it's so hard to do, because we can't infer behavior from a genome the way we can infer metabolic pathways. Okay? I think we'd all accept that. So why would we make the assumption that a microbial eukaryote was any different? This is a ciliate, and it's eating an animal. So this is a single cell protist eating an animal. If you sequence its genome, what would you actually know about its behavior in the environment, about its ecological position? Would you know that it had a great big mouth, that it could chase down animals and eat them? No, you wouldn't, unless you had a lot of natural history to inform that. So why do we make that assumption that microbial eukaryotes should be like other microbes rather than like other eukaryotes? Okay, so I'll get to HGT, horizontal gene transfer. This is the example I want to talk about. Horizontal gene transfer is the movement of genes between things non-sexually, so let's say between different species or distantly related species. It's a very important process in evolution. In fact, these are some just little snippets when HGT, or horizontal transfer, was first becoming well known. We have Darwin was wrong, cutting down the tree of life, all these kind of things came out. Because the idea was things aren't evolving in a tree-like fashion, they're exchanging genes horizontally. It's like, oh no, the sky's falling. And it turns out it is very important ecologically, and it's mostly known from bacteria, where there's lots of different ways that genes move, and when we sequence genomes, we find they have received lots of genes from other lineages. So there's lots of horizontal gene transfer, and it's very important evolutionarily and ecologically. A lot of this was actually driven by my PhD advisor, Ford Doolittle, and these are some of the pictures that are based on, or he drew, about what this meant for the tree of life. And so you see it's a tangled web with lots of things moving between different lineages, and you know, it's the question is it a tree or is it a web or blah, blah, blah. But what I want you to notice here is that in every case, the eukaryotes are over here, and over here, and over, oops, in the middle here, here. They're not a tangled web. You didn't draw any horizontal gene transfer in the eukaryotes. Why not? If you'd ask Ford about it, he'd probably say, I don't care about eukaryotes. Actually, I did, and that's exactly what he said. But most people sort of would have assumed that eukaryotes were just the same, or maybe even worse. There might be even more horizontal gene transfer. And I'll tell you why. Because of eating. So eating other cells. I can't stress enough how important that must have been to how eukaryotes evolved. When they started eating other cells, they were living in a world where everything was competing for metabolites, and suddenly their competition became their food. When you learn to take up another cell and digest it, which bacteria do not do, it put you on basically a new niche, a new plane of adaptation, where nothing was competing with you. So you could be really bad at it until other eukaryotes evolved and started competing with you. So it was a really big change to the way evolution happened. When eukaryotes originated, when they started eating other cells, I would argue it changed everything. So with respect to HGT, this has been identified as an important thing. But I'm going to argue that it's been interpreted exactly backwards from how it actually affected it. So again, Ford had this very influential theory called you are what you eat. His argument was, when eukaryotes started eating other cells by phagocytosis, they exposed themselves to an almost limitless pool of foreign genes. Every time they ate something, it's full of genes. They'd make their way into the nucleus, and if they were taken up, that was a one-way ticket. You kept it. If they weren't taken up, well, maybe you'll get the gene from your next piece of food. So he called it an evolutionary ratchet that had only one direction. Eventually, given enough time and enough feeding, you're going to replace all your genes even. And so this was a very influential theory, and it had to do with both just feeding, but also endosymbiosis, or when you feed on another cell, you take it up but don't digest it. And there the argument is even stronger. It's living inside your cell. It's dividing itself. Occasionally they burst. You get lots and lots of genes from them. So the eukaryotic nucleus should be full of foreign genes that you get from eating and endosymbiosis. It makes sense. It's a very intuitive idea. So it really took off. However, I don't know why this keeps doing this. Can you still read that? Good. I want to make a case that this is only one aspect of it. So feeding increases the chance of getting foreign genes. Yes, for sure. You're bathing your cell in foreign genes all the time. And those chance events are important because a whole bunch of them have to happen for a horizontal transfer to be real. You have to take up a gene. You have to integrate it into your chromosome. You have to express it. The protein has to be trafficked to the right part of the cell. Those are all complicated problems, and they all have to happen for a gene to be successfully horizontally gene transferred. And eating things certainly increases the exposure, the uptake of foreign genes. So this is probably important. But this also just overemphasizes these chance events, and it misses one very, very important aspect of this process, which is the gene also has to be necessary. Okay? I'm borrowing Jacques Monod's words here, chance and necessity. Because if the gene isn't fixed into the population, as in it's useful and you keep it and selection works on it, none of this matters. It could be so easy to pick it up, but if you don't need it and don't keep it, it doesn't matter. You're not going to keep it. Horizontal gene transfer requires both. You can see how this would work in bacteria. It's easy to see how bacteria should evolve by HTT a lot. But I'm going to ask whether it's true of eukaryotes too. And I think the answer is no. Okay, so in bacteria, remember they live in these metabolic networks. The ecology of bacteria is all about exchanging metabolites. Just bear with me with that. It's not totally true, but it's a big part of it. So they're adapted to a subset of the available nutrients. If they get an enzyme, this red thing, that can digest a nutrient that they have availability of but aren't currently using, then that could be selected for because now they have more food. Simple as that. So in a bacteria, that kind of change is really common and easily available through horizontal gene transfer. So basically you get a gene for this thing that can digest red stars. It's immediately advantageous because it's already evolved to digest red stars and some other bacteria, and you keep it because it's advantageous. It can sweep through the population very quickly and it fits you into the metabolic network more tightly, then it's advantageous, done. HTT is a very powerful source of new variation for selection to work on in bacteria. This is why it's so prevalent in bacterial genomes. But let's think about eukaryotes and eating things for a second. First off, would that work in this eukaryote? Okay. Can it acquire more food by HTT? No. It's not going to get smart genes from eating smart food and then become more smart and get more food, because that's not how it works. It wouldn't happen with a cheetah. Cheetahs don't hunt down and eat fast things and get their fast genes and swim and then run faster. No one would ever accept that. It's just ridiculous. They're fast runners because they have developmental programs that have changed the structure of their body that gives them the ability to run more quickly, right? So again, I'll ask, why would we assume this about microbial eukaryotes, which we have assumed, and not assume they're more like other eukaryotes? Because these are also creatures of their behavior. They're hunters, they swim, they shoot poisonous harpoons, they sense light and adapt to it and swim faster, swim slower, they have bigger, smaller mouths or whatever. They're not going to evolve the same way. So here's what I mean. So we have the bacteria up here. So down here, let's imagine a eukaryote that's eating bacteria. And it has a small mouth, just to make this simple. It's just eating the blue bacteria because the purple ones are too big. What kind of adaptation is going to change that? If you have a cytoskeletal change where it's remodeled and your mouth is a little bit bigger and now you can eat the purple bacteria and the blue bacteria, you've just got more food. So that's sort of equivalent to the change up here, but it's a different kind of change because it's not the kind of change you're ever going to get by horizontal gene transfer. It's the kind of change that you're going to get from things like epigenetic changes, point mutations in regulatory regions, expansions and contractions of gene families, stuff like that. Not horizontal gene transfer. It would never give you that kind of change. And indeed, these are exactly the kind of changes that we do see being very important in eukaryotic genomes. Just the same as we accept in animals all the time, it's true of other eukaryotes as well, microbial eukaryotes. And in fact, HTT tends to be fairly rare in eukaryotic genomes, 10 to 50 times less than what you see in bacteria. And so it holds up. There are exceptions, and they're fascinating exceptions. The best one is fungi. These are just clips from papers. There's loads of HTT in fungi compared to other eukaryotes. But that actually makes sense. If you think about fungi, I'm sorry, I'm going to make fun of fungi now. I actually like fungi, but fungi are really boring structurally and behaviorally. They're really exciting metabolically, but structurally and behaviorally, not so much. I mean, yeast it's a easy target, sorry. Yeah, they float around and they absorb nutrients. They eat by saprotrophy, osmotrophy. They're basically eukaryotes that have turned into bacteria in terms of their metabolism and ecology and the strategies that they survive by. So it actually makes sense that they also acquire genes by HTT because they're immediately selectively advantageous and they help that fungus thrive. Another interesting case is the oomycetes, which people used to think were fungi because they kind of act like fungi, and it turns out they also have acquired a lot of genes by HTT from fungi, probably to adapt to that lifestyle. And we do see that. There is HTT in eukaryotes, and it is ecologically important. There just isn't very much of it, and it tends to be things to do with metabolism. So, just to summarize that, so feeding and phagocytosis, which were thought to be engines of HTT, are actually probably increasing the chance of getting HTT, but the act of eating that way also undercuts its necessity because you've changed the way you're evolving by becoming a hunter that makes HTT a poor source of variation to be acted on by selection. And so it actually ultimately decreases the importance of HTT in eukaryotic evolution compared to prokaryotes. So I'm not saying it doesn't happen, it does, but it's rare and it tends to be targeted at certain kinds of processes like metabolism. And in organisms that have changed their metabolism, a lot of it becomes very important, but it's not very common. So I want to then focus on symbiosis because this is another case where it's been made to be really important, where the role of HTT is well studied in endosymbiosis, and I want to cover this a little bit. So this is a mitochondrion, of course. It's an organelle in the cell that's arisen by endosymbiosis. It was a bacteria that was taken up and genetically incorporated into the cell, and of course chloroplasts and plastids is the same, with cyanobacteria being taken up. So these have played an outside role in our view of eukaryotic evolution. I want to talk about that. The first thing I want to mention is this process here, endosymbiotic gene transfer. So this is an important concept, and it was actually first pointed out in 1967 in a very much forgotten paper that came out the same year as Lynn Margulis's famous paper about endosymbiosis. But this paper came out the same year and was actually a little bit more thorough, but everyone's forgotten about it. And then it was really, this concept was really elaborated by Whedon much later. And the problem was that if you look at a mitochondrion and you look at its genome, or a chloroplast in its genome, they don't encode enough genes to work. So where are the genes? I mean, we know this now, but at the time it was a real mystery. Where are the genes coming from? They thought, well, they must come from the nucleus. But if these are endosymbiotic bacteria, where did the nucleus get the genes? And Whedon in particular very, very smartly recognized, well, they probably came from the symbiont. And they moved into the nucleus, and somehow the host targets the proteins back to the organelle. And that genetic integration, the movement of genes from the symbiont and the targeting of the proteins back, is what we really now use to define an organelle over just any old bacteria that lives in the cell. And so this was a very important concept, endosymbiotic gene transfer. And parts of it are still right today, but parts of it are wrong. So I want to go through a couple little simple things here. Basically, this is the concept of endosymbiotic gene transfer. You have a little endosymbiont living inside a eukaryote, genes are moving, that's arrows, and proteins are targeting back, that's these dots, okay? So this is basically the nut of it. Genes moved, the genes are expressed, targeted back to the organelle. That's EGT. So originally, when people conceived of this, here's a pile of genes that all transferred from the symbiont, and the proteins are all targeted back to the symbiont. We imagine them to be one and the same. All the genes that moved, the proteins are targeted back. That was how Whedon saw it, say. But over the last decades, this concept has shifted a little bit. And there's been two different kinds of genes that have all been called endosymbiotic gene replacement, and it's made it massively confusing. And so I want to try to introduce two different terms to separate these three things because they're very different things. So the idea here is you had a bunch of genes transferred to the symbiont, or from the symbiont, you have a bunch of proteins targeted back, but it's only a partly overlapping cluster of genes. Mostly overlapping, but not quite overlapping. So you have some genes over here that were transferred but aren't targeted. And then you have other proteins over here that are targeted to the symbiont, to the organelle, but they didn't come from it. They came from somewhere else. And so I want to try to introduce new terms. I'll call this hetero-endosymbiotic gene transfer, meaning that it's, it's a gene that transferred from the symbiont, but it goes somewhere else. Now, the protein. And these ones are proteins that are targeted to the symbiont, but the gene came from somewhere else. Okay? And we're going to call those hetero-endosymbiotic protein targeting, just to try to keep these three things separate. Okay? So if you think about how organelles originated way back in time, if you look at a textbook, you see a diagram that looks something like this. And you might think of this as like a cellular indigestion. You ate something and you choked on it. So we got an organism that's eating bacteria and digesting it fine, but then it keeps one. It doesn't digest it. It sort of shunts over here and doesn't get digested. And then, as the theory goes, it starts moving genes into the nucleus. Those genes can't be targeted back because there's no targeting peptides, there's no targeting machinery, and so the proteins are just kept in function in the cytosol. And then a targeting system evolves, and more genes move, and then their proteins can be targeted back. And so you end up with something like this, where you have a bunch of genes moved into the nucleus, a lot of them are targeted back to the organelle, and some of them aren't. Maybe a lot of them aren't. Okay? So this is kind of the mostly the models are vaguer than this, but if you dig into the people that are really proposing serious models, this is mostly the order of events that they say. And this has been a big business in science. There's been a lot of papers about this. There's been a lot of papers looking at the fate of genes that come from the endosymbiont. So for instance, this is a very influential paper up here that said that thousands of genes from the Arabidopsis genome were actually derived from the cyanobacteria that gave us the plastid. But only half of them, at most, were actually targeted back to the plastid. The rest of them function in the cytosol. Same claim was made in yeast, that a vast proportion of the genes in the yeast genome came from the mitochondrial symbiont, but don't function in the mitochondrium. So in other words, that the symbiosis led to a huge influx of genes, thousands of genes, into the nuclear genome, giving new functions to the eukaryote that didn't have anything to do with the organelle. So a massive influx of horizontal gene transfer due to eating and symbiosis. And this has one other important implication. So here's the model. What happens if you lose the organelle? If it disappears, you've still got a ton of genes that came in with it. And this has been very influential because this has been used, the assumption that this big pile of genes must exist has been used to test for the past presence of an organelle that's been lost in several different ways. So this has been put best here, and it's called a genetic footprint of an organelle. The idea that it would leave behind a genetic footprint of itself after it's been lost. And basically, this is what I'm calling hetero-EGT. It's all these proteins that originally came from the symbiont, but now the symbiont's gone and the proteins are still there, giving a little clue that the organelle used to be there and has now been lost. Very influential thing that's been used to test many, many hypotheses and sort of led to a lot of big-scale ideas about eukaryotic evolution. But guess what? we've never actually tested that those genes even exist, let alone that they should still be there today, as a basis to test another hypothesis. So it's sort of like a meta assumption. It's like an assumption based on an assumption. And that gets really creepy for evolutionary biology. So I can test that assumption with a couple of systems. This is a massively complicated scheme showing plastid evolution, where we have a cyanobacteria got eaten by a eukaryote and led to many of the algae and plants we know today. Well, then they themselves have also been eaten by other eukaryotes several times, leading to more of the algae that we know today. And down here, it's happened again. So some of the algae that have these already complicated plastids are eaten again by another algae, leading to more complicated things. So this is called primary, secondary, and tertiary endosymbiosis. These things are all pretty recent, and so they're actually pretty easy to study things at the genetic level and know what to expect, because they come from well-known groups of organisms, and we can make trees that are fairly reliable. So there's two cases where we can say really concrete things about what happened. The first is a case of a secondary plastid in euglenids where a green algae got eaten by something. The other one is a tertiary plastid where a particular kind of red algal containing algae got eaten by another red algae containing plastid, leading to permanent fixation in the cell. So this is a little complicated, so I'll walk through it. This is the case with the haptophyte. And this is a system that Becky Gast at Hui developed, and it's an amazing system for evolutionary biologists. So we have up here a couple of algae that are well known to have eaten something called a haptophyte and kept its plastid. But what she showed is they actually ate different haptophytes. So the plastids originated here and here separately in related hosts with related plastids, but slightly different lineages of haptophytes. So they happen twice, not once. And then she found something called a kleptoplast is related to all of these. And this is something that picks up a plastid from a haptophyte, but doesn't keep it forever. It eventually digests it and goes and eats another one. So it's a temporary plastid. And so this system has many independent origins of this event that we can look at as evolutionary biologists and see what the ancestral states were like and see what the steps were like. If you sequence everybody's genome, that is. And so that's what we did with Becky. And we found a couple interesting things, which is, first of all, the protein targeting system that should have evolved last actually evolved first. It evolved before the organelles were even fixed. So they all share the same targeted proteins even before the organelles are there. So that may seem like the cart before the horse, but bear with me. We also found they've got lots of EGT. They have lots of this hetero-EPT. Remember, that's proteins that go to the plastid, but they're not from the plastid. They're from something else. But they're all targeted to these different plastids. It didn't have a single gene from the endosymbiont that wasn't targeted to plastid. Not a single one. So it had this other thing living in it and turning into an organelle, and it took 0 genes from it except for the ones that it targets to the plastid. So this thousands of genes that should be there is 0, which is interesting. Is this normal? Is this just a freakish thing, or is this how endosymbiosis works? Happily, there's another case where you can test it. This is the euglenid, and we have the fixation once of a plastid in this group, but now another group has found a kleptoplast that's sister to it, very, very similar. So a temporary plastid, but it's also a green algae, but a different green algae. And it's down here at the base of this group. So it's like a really interesting, fascinating glimpse into this process in play. And if you sequence all these genomes, you find exactly the same thing. You find the targeting evolved first. They target the same genes into the plastids, even though they're different plastids. We find lots of EGT, lots of foreign genes being targeted to those plastids, and we find 0 genes from green algae in the host lineage that don't have to do with the plastid. So this is an important process, this is an important process. This is a phantom. It's something that we thought was really important that probably doesn't even exist. So the answer is yes. I think, at least in these two cases, every single symbiosis might be a little different, but I'm saying the only 2 that we can actually look at in any detail show this pattern exactly the same. So what about other things? Turns out there's all sorts of bacteria that have been genetically integrated with their host that we don't call organelles. They're not mitochondria, they're not plastids, but there's lots of them cropping up. The most famous is this amoeba that's got a cyanobacteria in it that's basically like another plastid, but it's not related to the same lineage. It's a Synechococcus, Synechocystis. And it's been studied fairly well. There's also this Angomonas, which is a beautiful newly discovered system being developed. It's a bacteria that lives inside a trypanosome, obligate symbiosis. And then there's lots of insects with obligate bacterial symbioses, where the bacteria live inside their cells and provide them with amino acids. That's pretty common for sap-feeding insects. Turns out all of these cases are genetically integrated. So there's proteins that are encoded in the nucleus of the host that are targeted to these bacteria. And in all cases, there is foreign proteins that are not from the lineage of the bacteria. They're just from something else, and they're targeted to that bacteria. So that's my hetero-EPT. EGT is not always the case. There's some cases where none of the genes from the bacteria have moved to the host. The only exception is Pollinella, and none of them have any genes from the bacteria encoded in their genome that don't get targeted. So it's exactly the same pattern. So basically, this model's wrong. We should ditch it. We should delete it, rip that page out of all the textbooks, because it really doesn't explain anything. So I'll give you an alternative, just one alternative to think about. So imagine you have a eukaryote that's eating and digesting stuff. That's not so hard to believe. That happens a billion times a second. Now imagine that it starts what I called sort of delayed digestion or farming, where it just keeps things in its cytosol for a while because they're kind of more useful than just digesting them. Maybe they're making a vitamin or whatever. So this is also fairly common. So you just delay digestion for a little while, extract nutrients from it, and then digest it. So what would promote extending that delay, making it longer and longer? It would be getting stuff from it more efficiently, right? And so you could target proteins to that thing to help that would be beneficial. So you could target transporters to suck nutrients out of it instead of just letting them diffuse. Or you could target proteins that stopped it from replicating unlimitedly, because that's dangerous. So you control your symbiont, that's what I'm saying. You target proteins to control your symbiont. So protein targeting evolved first because it didn't evolve to target proteins from the symbiont back to it, because that's kind of stupid. Protein targeting evolved to take control over that symbiont so you can abuse it even worse and get more from it, right? So that would be the impetus for delaying digestion, is protein targeting to enhance your power over the symbiont. This, though, initiates a ratchet where once genes do move from symbionts, now there's a targeting system already there. They just have to acquire a little peptide and boom, they go back to the symbiont. And so as you're digesting symbionts, you're probably picking up more and more proteins with every round of this that are then targeted back to the next symbiont. Not to that one, you just digested it, but the next one would get a protein targeted to it that the last one didn't. So this is a ratchet. Eventually, you fix it. I don't know why. Maybe it was a mistake. Or maybe, you got a really good symbiont or whatever. I don't know. I don't have any explanation for this, but let's say it did get fixed once. Well, the genome's already got the targeting system. It's already set up to target stuff. It's also got a whole bunch of genes all ready to go that are targeting to this thing. And it opens up a new ratchet where you can now pick up genes from your now permanent symbiont 2. Okay? So if you think about it, these are the EGTs that we know and love. They're all there. We know that the mitochondria has lots of proteins from the mitochondria targeted back to the mitochondria. That's great. But it also explains those hetero-EPTs, the foreign proteins that we find are targeted to all the organelles, are the ones that it didn't pick them up later, as they've been sometimes interpreted, including by me, but it actually might have picked them up before the organelle even evolved, just through this cyclic process of picking up organelles and farming them for longer and longer. So yeah, EGT is predicted by this, hetero-EPT is predicted by this. This isn't predicted by anything, but I have to say in this model it's also not essential. There's no reason to expect these to exist. Because remember, eukaryotes don't find these genes very useful. They're not going to get genes by HTT because they're not very useful, and they're not going to get genes by endosymbiosis either because they're also not very useful for the way eukaryotes evolve. So they get genes related to the organelle because those are useful, But the rest of them, they don't get genes from HTT anyway. Why should they get any from an organelle? Okay, I'm just going to finish with a little philosophy. That's good timing. Because it raises an interesting question. If you sequence a genome, can you tell the difference between an organism that lost a plastid, okay, or a mitochondrion, from one that never had one? And the answer is no. We can't. Because we should assume if there's none of these if the organelle is not donating thousands of genes to the nucleus that have nothing to do with the organelle, if you delete the organelle and all its functions, it leaves nothing behind. There's no footprint, right? Because there never was a footprint to be left behind. This is a little troubling when you think about hypothesis testing, because we can find evidence that there once was an organelle. It's possible that there could be a tiny little footprint just by chance. Sure, that's possible. But you could never have evidence to falsify the past presence of an organelle, because you can't assume that they would have left a footprint, right? And so what would Karl Popper say? He would say that's not science if you can't falsify it. And so all this speculation about past organelles and looking for this footprint is actually beyond science, because it's not falsifiable. And to prove the point, we're actually getting to the point now, this is very rare, but we have found organelles that are totally lost. And it is hard to prove because they have to be in a clade where you can demonstrate conclusively that their ancestor had an organelle, and then you have to prove conclusively that they don't still have a tricky little hard-to-find version of it, because a lot of them have tricky little hard-to-find organelles. But it has been done now. Once for the mitochondrion, this fascinating case, oops, that's the wrong one, this one. Fascinating case where this organism has completely lost its mitochondria, which is a trick that no other eukaryote has been able to repeat. And then there's quite a few which have lost, where we have good evidence, or really good evidence, that they've lost a plastid. These are mostly parasites, but not entirely. There's a couple free-living things which have lost. Photosynthesis is easy to lose, but then losing the plastid is very, very difficult. Almost never happens. But now we've got multiple independent cases in different groups where the plastid has been lost. And if you look through these genomes, for the footprint of the mitochondrion or the plastid, you find nothing. So when they lose a plastid, they lose it. There's nothing left. The biochemical pathways don't move into the cytoplasm. They just get deleted because they don't need them anymore, which is why I lost the organelle in the first place. So this kind of reinforces this point that you can't assume that this footprint exists. You can't use it to test anything. And we're totally misunderstanding the importance of endosymbiosis in eukaryotic evolution, because it is not contributing thousands of genes to the nucleus that don't have to do with the organelle. It's important enough as it is. It didn't really need to have that extra add-on, but that add-on has been this red herring that's actually causing a lot of trouble in misunderstanding evolutionary events at the biggest level in eukaryotic phylogeny. Okay, I'll just end. I want to emphasize a couple things. So phagocytosis, one of my favorite processes, it probably increased the chance events that would favor things like getting genes by horizontal gene transfer, but by its very existence, I would argue, it also undercut their necessity, and therefore we should have expected in those organisms that HTT might be rare, instead of thinking that phagocytosis would make it more common. So I think we got that backwards. In the case of organelles, basically there's really no evidence at all that EGT, that genes from the organelles contributed to the nuclear genome beyond things to do with organelle function. And I think we have to stop treating that as the null hypothesis, because we keep finding it doesn't happen. The null hypothesis at least should be that it doesn't happen until you can prove it does. But right now it's being treated the opposite way. And then this has this interesting implication that I find fascinating, that we can't tell the difference between something that lost an organelle from something that never had it, unless you have a really good phylogeny and you know exactly where it goes in the tree and you know that all of its ancestors had that organelle. That's the only way you can tell. The genome won't tell you because there's no footprint. All right, I want to thank lots of people about this. I had lots of discussions on the the subject of HCT and phagocytosis particularly. And these are the people that I talk to, and also thank my funding. And again, just to end by thanking the organizing committee and my host. It's been grueling but fun so far. I've had so many cool discussions. I'm a little bit brain dead, so I hope I wasn't just up here going blah, 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 because I'm not sure. Thanks.
Questions? Yeah.
So concerning that diagram that you had up earlier where you proposed a new like nonlinear model, I noticed there was sort of like an addition of like this arrow of proteins that were used to manage the new organelle, or the, yeah. And isn't that a kind of footprint, or like, how is that different? Yeah, like then there's like another pathway that it develops to manage that organelle that it learned from like the previous one. Is that what you're saying?
Yeah, good question. So the question is whether these proteins, the targeting system, or the first proteins targeting to it could act as a footprint. So the targeting system itself would not, because once the organelle is gone, it doesn't need to target. So those proteins always disappear. Ticks, tox, stim, stoms, and things that we know from current organelles. These proteins that are to do with control, they might be like, you can imagine a dynamine ring that controlled the replication of an organelle or something like that. They would also be lost because they're tied to the organelle function. And so the only things that are going to get kept, which I haven't got on here because I don't actually believe they probably exist, would be something that it got from some fixed organelle that then functions somewhere else, completely independent of organelle function. So those proteins are really important, and they don't have to do with oxidative phosphorylation or photosynthesis or whatever, but they're tied to the organelle function. And if the organelle is lost, they don't have a function anymore, and they'll probably also be lost. Does that make sense?
Thanks. So if most of these genes that are getting transferred to the nucleus are getting targeted back to the organelle, it's obviously a bit roundabout. So like, is there a consensus on what the major selective advantage is for transferring them to the nucleus in the first place? Does it have to do with like advantages of sexual reproduction or something else?
Sure. There's actually been a lot of discussion about why do you move things to the nucleus anyway? So, and I think there's probably different answers for different proteins. So you mentioned, is it advantageous to be under sexual selection? Maybe. And this is a weird place to be in an organelle. It's a very funny, population genetics of organelles is very bizarre, and their genomes get really weird, and the gene sequences get really weird. So yeah, there might be some advantage to keeping important genes in the nucleus. But then if you look at the biochemistry, most of the really core genes at the reaction centers are the ones that they keep in the organelles, not the other way around. And that's been argued to be because they need tight regulation or something like that. I tend to think it has a lot to do with the fact that those membrane proteins are really hard to target because they have to cross a bunch of membranes without getting stuck in them, or a combination of all of the above. But it's a good question because that's actually, I'm turning your question on its head and saying, why did you keep anything in the organelle? The question of why things moved to the nucleus, it might have just been because they could, and it is a ratchet, because this is a one-directional thing. It's possible some of them move neutrally. So you have a situation in plants where very closely related plants, some of them have an organelle encoded gene, and then some sister group has the same gene in the nucleus. I suspect that has no functional reason whatsoever. It's just in that one it moved and it can target back and it still works, so it makes no difference. So again, I think it's a bit of a combination of different things for different genes. Some of them might be better in the nucleus, some of them definitely are better in the organelle, and some of them probably don't matter. But I don't have evidence for that. Sorry.
I'll just make a quick announcement to say that I'm monitoring the Q&A on Zoom as well. So if any of the Zoom participants have questions, feel free to post them there and I'll ask them. Any other questions in the room?
So it's a question about the loss of organelles. Do you think we're very good at identifying which things are highly derived organelles, like the MROs or the apicoplast or something like that? Do we know most of the time?
Yeah, that's a great question. And honestly, I would have said 15 years ago, no way. We're terrible at that, but we're getting way better at it. So the cases I showed are restricted to cases where we have basically, let me back up. If you try and find an organelle with microscopy, you know, in a way that should be the standard. Well, there it is, I see it. But these MROs that you mentioned and the apicoplast, the derived plastid of apicomplexa, which I'm going to talk about tomorrow, it's a big plug, they're hard to recognize because they're really just reduced bags with no structural, morphological similarity to the things that we see in textbooks. So they're really hard to find, and they escaped our attention for, in the case of the apicoplast, decades. But now we're good at it, and the things that I listed are only ones where we've sequenced the genome. So you sequence the whole genome, and things like the targeting, the ticks and the tox are gone. Everything to do with mitochondrial function's gone. The mitochondrion is particularly easy because almost all mitochondria that have been highly reduced but kept are making iron-sulfur clusters. That's the essential function of mitochondria. You know, we're a bit biased because we're animals, so we think oxidative phosphorylation is really important, but really, who cares? But that process is really important, and they've only managed to lose it because they acquired a bacterial iron-sulfur cluster assembly machinery by horizontal gene transfer. And so if you go back in their ancestor, you find all of a sudden there's this SUF system that can replace the ICS system. And then there's aerobic relatives that have both. And then there's an anaerobe evolved, and having lost the need for aerobic respiration, it was free to lose the whole organelle. So it was actually set up to do this before it became an anaerobe, if you see what I mean, just by chance. And so the same thing tends to be true in the plastids. Things lost photosynthesis, and then a tiny subset of them have gone that extra mile and lost the whole plastid, but it's very rare. So I'm being very conservative. Those are usually things that are well studied with microscopy, can't find anything, sequence the whole genome, there's nothing left. And they've got really well studied relatives that have lost photosynthesis, say, and we know what pathways they still have, and this thing doesn't have those pathways. So that's kind of, it's a fairly high standard of evidence, but we could be wrong sometimes, but I'd be really shocked if we were wrong every time.
There's a question from the Zoom audience, from Luca Kamai. Could endosymbiosis between two eukaryotes lead to nuclear fusion?
Well, there has been lots of endosymbiosis between two eukaryotes, and I think that, I mean, in two distantly related eukaryotes, I really doubt it. And I'm going out on real thin ice here because I don't think about populations very much. But I believe it's definitely more likely if you had two very closely related, you know, plants or fungi or things where there is cross-hybridization sometimes demonstrated that they could fuse nuclei, but I would guess they'd have to be extremely closely related. Yeah, but there's no evidence of it happening between distant relatives that I'm aware of.
Thank you. So I'm wondering if you could talk a little bit about the difference between the kleptoplasts and the farming phase. Are kleptoplasts just something we're observing that happens to be during the farming phase, or maybe it's stable, or I was wondering if you could elaborate.
Yeah, so kleptoplasts basically are a really obvious, highly advanced form of farming. Sure, yeah, because they're kept for, the one that I worked with most was the dinoflagellate that ate the haptophyte. That thing eats a haptophyte, and it reduces it down, it digests most of it, including the nucleus, and it just keeps the plastid, and it can keep the plastid for, I think it was 3.5 years. And that's when the incubator broke, so it was probably longer. Becky's like a genius at this stuff. She grows, it's a psychrophile, so it grows really slowly at very cold temperatures. It's from Antarctica. And yeah, it keeps it for a very long time, which is way longer than the half-life of all those proteins. So it's definitely, you know, an active, targeted, it looks like an organelle. and then it just eats it and goes off and eats another haptophyte and gets a new one very, very infrequently. And there's other things that do this too. There's a famous animal that does this, the Alicia, the sea slug, does the same thing. It moves the plastids out to its epidermis, and it just sits there and changes shape and photosynthesizes. This is a very elaborate form of farming, yes. There's things that farm, I'm getting off topic because I love this stuff, but there's extracellular farmers too. There's ciliates that farm bacteria on their backs, and they make these big flaps, and they to maximize their surface area, and then they just engulf the bacteria one at a time like they're picking fruit, and they just grow the little monoculture of bacteria on their back. That's farming too, but it's extracellular, so you'd expect it would have different impacts. But yeah, kleptoplasty is just a really clear case of farming, like a really obvious one. Yeah.
Thanks. So most of the discussion about transfer seems like it's been sort of from the perspective of the eukaryote. And I'm wondering if you can say a little bit about this from the perspective of the organelle or the proto-organelle. I mean, like some of these transfers putatively could happen because they're advantageous for the organelle rather than for the eukaryote, and then what that sort of means. Maybe that doesn't happen, and that's why they're all lost. I don't know. Maybe you could say something about that.
Actually, I really like that way of thinking, because I find almost we're so animal-centric that when we think about symbiosis a lot, like when you think about microbiome research, it's like one of my things, is that we always think about it from the perspective of the animal. What's the benefit, blah, blah, blah. I've been asking for years, where did microbiomes come from in an evolutionary sense? Everybody looks at them physiologically or generationally, but where did they come from evolutionarily? Almost certainly the bacteria were running it. They were just good at living in animals, and there was a guild of them that invaded animals and were good at it. And that's where microbiomes came from in an evolutionary sense, and there's evidence to support this. So why wouldn't I say the same thing about this? It's because actually there's a couple different kinds of symbiosis. When you look at bacteria living inside eukaryotic cells, there's exceptions, but a lot of them can be divided into two. The one is like what you're talking about and what I just mentioned with microbiomes, and we call them professional symbionts because they're really good at getting inside a eukaryotic cell and not killing it and just living there and kind of sliding under the radar. So they're probably a little deleterious, but no more so. I was making a joke about it. It's like someone sleeping on your couch. Like you want to get rid of them, but whatever, right? Because they're not doing them that much harm. That's really common. So Rickettsiales, metachloraceae, stuff like that. So I don't think these are those. I think they're more like the other class of symbionts, which are the ones where the eukaryotes calling the shots. That's the bacteria in charge, and their genomes are different. The ones where the eukaryotes in charge, they're in trouble because they're almost all going to go extinct. The rare exceptions are when they get stuck, but their extinction's by far the more common situation. I'm sorry, there's a long answer, but there's some really interesting cases now where you have a bunch of related eukaryotes that have related symbionts, and it looks like an ancient symbiosis. And when you sequence those symbiont genomes, you realize they're all different derivatives of different free-living bacteria. In other words, the eukaryote has been for centuries or millions of years picking up related bacteria, basically enslaving them and using them to death. And sometime before they die, they pick up another one, and then they go extinct. That's actually a very common outcome to symbiosis, where your bacteria getting stuck by a eukaryote. And it's bad for the bacteria. And I think these are like that, except the organelles got lucky and escaped and became permanent slaves. Do you call that luck? I don't know. But I don't actually think I don't think this is good for organelles. Does that kind of get at what you're saying? Yeah, sure. There's a lot to talk about here. It's a really interesting topic.
If there's no other questions, I have one. So when you're looking for the evidence of past organelles in lineages that have lost the organelle, and you can't find it, is it a possibility that we don't have yet a complete enough understanding of organelle proteomes and the biodiversity of them such that the signal that you're looking for is kind of cryptic or hidden, and we need a better map of, say, the proteomes of the ancestor of that lineage that's incomplete at this point? Or
Sure. I'd give that's definitely a yes at a theoretical level, but I think maybe at a practical level it's not that likely. Because you'd have to, and we've been talking about this, that there's a bunch of proteins in the organelles that are hard to explain. So, but you'd have to have lost all of those sort of flagship organelle systems that maintain the organelle in a bunch of related lineages. So if we take the apicomplexans, which someone mentioned, we know that they're plastid. These are obligate intercellular parasites, like the thing that causes malaria, but they evolved from an alga, and they maintain a bunch of biochemistry in their plastid. DOXP pathway for isoprenoids, fatty acids, heme, iron sulfur clusters. So every other group or every other member of that group has some subset of those things, and then you get to an organism that doesn't have any of them, and you can't find the apicoplast. And then now we've done taken Loppet data from toxoplasma and applied it across this diversity, and we find they've also lost everything to do with organelle maintenance, everything to do with the organelle genome, everything. Even the orphs that we don't know what they are that localize in toxoplasma are gone in these things. And so I actually think you're right. I think that's the kind of bar we need to set ourselves, and for most systems we don't have that kind of data. But I think in that system we're very confident.
any other questions? All right, if not, let's thank Dr. Keeling again for a wonderful talk. Thank you. Thank you so much.